The second messenger, cGMP, is an extremely important target for disorders such as heart failure, atherosclerosis, and cardiac hypertrophy, disorders affecting millions. The sGC protein is a site that converts GTP to cGMP, a small molecule controlling vasodilation. Currently, very few to no therapies target the sGC-cGMP pathway due to many unknowns about the protein structure and qualities. cGMP can not only treat cardiovascular disorders, causing up to 31% of world deaths worldwide, but also likely have a role in cancer regulation, major depressive disorder, and more. Understanding the sGC-cGMP pathway with evolutionary tools is of the utmost importance to target these deadly illnesses. This study seeks to first categorize the protein through models created from databases and deep learning software. Then, multiple protein sequence alignment is used to determine that the HNOX domain of the protein has a high concentration of conserved residues, the parts of the protein that stay constant across evolutionary lineages and are therefore most important for the protein function. Finally, a blast genetic sequence analysis is done to compare sGC proteins such as hemoglobin, and it is determined that sGC is a druggable target. After that information is collected, a virtual screening of drug candidates is done. After reviewing sGC structure, it is visible that it is likely a membrane protein due to the helix. The HNOX domain was first proposed as a selective membrane may prevent small molecules from passing through unless systems are put in place. Additionally, conserved residues are concentrated in the HNOX domain, so it would be confirmed that the domain is functionally important for sGC. Finally, the HNOX domain contains a heme site, which is important for signal transduction. The only issue is if hemoglobin and sGC have similar structures, as small molecules may harm hemoglobin, a protein vital for carrying oxygen and carbon dioxide to and from tissues. By superimposing the two structures with a multi-aligned viewer, it is found that the heme sites occur in different locations, and in fact, targeting it would not be damaging. The HNOX domain has some pi-pi stacking, one charge center, and many hydrophobic reactions, with the outer lining covered with hydrogen bonds. The domain has only one salt bridge, and it is on the edge of the grid box. Salt bridges may destabilize protein structures, which can make it vulnerable to aggregation and degradation. Notably, most pi stacking was perpendicular and not parallel. For the genetic analysis, genes that co-express and with a high Spearman's correlation are looked at as they may have similarities or contribute to MMD. Both genes RNF213 and GUAY1B1, which likely influence sGC, were analyzed. For RNF213, HELZ2 has a Spearman's correlation of 0.769. It is a co-actuator for peroxisome proliferator activated receptor alpha. Additionally, OAS3 regulates the expression of chemokines and interferon repressive genes. Some other co-regulators were DDX58 and ZNFX1, which have a hand in immunodeficiency, hyperinflammation, and parotid gland cancer. For GUA1B1, MAP9, microtubule-associated protein 9, has a Spearman correlation of 0.746 and is required for spindle function, mitotic progression, and cytokinesis. GLRB, glycine receptor beta, had a Spearman correlation of 0.703 and encodes the glycine receptor, a neurotransmitter-gated ion channel. Finally, the PDGFC, platelet-derived growth factor C, has a Spearman's correlation of 0.689, and diseases associated with this gene are Milker's nodule, age-related macular degeneration. By gathering this important structural information, drug candidates are proposed after conducting a virtual screening of thousands of ligands. Drug candidates, namely C15H24N4O, C9H11NO, and C16H20N4O4, were identified. The drug targets identified can move on into lab testing on animal models to be developed further into medications, as there is a lack of FDA-approved treatments. Finally, a combination of invasive treatment and medication can be used to reduce the chances of stroke or high blood pressure. As soluble guanylyl cyclase contributes to a variety of conditions, from hypertension and MDD to even cancer, further research in drugs targeting soluble guanylyl cyclase would be imperative. Thank you so much for listening.